Welcome to the 3D'omics Sneak Peek series, where our partners introduce their work. Today we will hear about Jenny Merkesvik from the Norwegian University of Life Sciences.
Hi, my name is Jenny Merkesvik and I'm a PhD candidate in bioinformatics at NMBU, that is the Norwegian University of Life Sciences. I joined the 3D'omics team in August of 2022. That means that I've been a part of the project for almost 2 years now. As I mentioned, my PhD is in the field of bioinformatics. This means that I work with huge amounts of biological data and I try to discover patterns and trends within them. I do this by using tools such as statistics, plots and visualizations, and of course the huge library of already known knowledge that we have on biological pathways, on genes, on proteins, and so on. I like to think of bioinformatics as kind of a detective work mixed with a bit of organization and a good dose of creativity. And in the setting of 3D'omics, these skills are really useful. The 3D'omics project generates loads of biological data. We've run several trials already, spanning both swine and poultry systems, and from each of these trials we've collected hundreds and hundreds of samples. From each of these samples we can generate a wide array of different types of biological data. For example, information on the animal themselves, in terms of what sort of genes they are actively expressing. We also get information on metabolites, meaning what sort of molecules and compounds are present within specific areas of the animals at a given time. And we also get information on the microbiomes of these animals, meaning the sort of bacteria and other microorganisms that are present within the animal at a given time, and also what sort of genes they are actively expressing. As a bioinformatician, I view all of these different types of data sources as layers, and each of them tell one side of the story on how this animal and its associated microbiome work together as one functional biological entity. Then by exposing this entity, meaning the animal and the microbiome, to different conditions, like in one of the trials where we fed half of our pigs with a fiber in addition to their regular feed, we can then see whether any of these data layers show that the animal and or the microbiome seem to respond or change their activities as an effect of this change. Then, depending on what sort of data changes we are seeing, we can try and determine whether, if, and of course also how, the animal and the microbiome is kind of cooperating and interacting in order to overcome any potential challenges or changes in their diet or in the environment, and in some way try and improve their health and performances. We can then use this insight and talk with industry representatives to try and improve animal and feed production practices in favor of better animal welfare, of production efficiency, and of sustainability. My tasks within 3D'omics are related to the 3 swine trials that we have run in the project, and what we in 3D'omics refer to as the conventional data that have been generated from these animals. This means that I am not working with the novel microscale technology that other 3D'omics partners are developing and working with, but rather exploring what we can learn from these animals and these trials using methods that are already existing and kind of serve as the groundwork or golden standards within the field already. This means that 3D'omics in some way will have 2 parallel investigations of the same animal trials. First this 3D'omics novel technology, and then on the other side what I'm focusing on these conventional methods. In some way that means that we get to investigate our animal trials and all of these different microbiomes and hosts and how they interact in 2 different ways. But it also means that at the very end of the project we get to compare these 2 approaches to see how this novel 3D'omics approach compares to what we are already doing within the field. So, for the past 2 years, my tasks have been to learn about how biological data from the host and from metabolites and from the microbiomes can be analyzed, and also try and see how we can fit all of these jigsaw puzzles together to try and create a better representation of the entire system as a whole, meaning the animal and the microbiomes as one united biological entity. In addition to these bioinformatical analyses, I've also had the pleasure to contribute in collecting the samples that these data have been generated from. Firstly, I was just helping out collecting some samples, but then at the very last trial that we had at NMBU, I got to coordinate the collection process. And while this was a very different kind of set of tasks than what my usual office job includes, it was a really cool challenge and an opportunity I doubt I would have had if not for 3D'omics. I'm really eager to soon be able to look at the data generated from these samples, and hopefully they'll show us some really cool patterns and help us learn even more about the mystery on how host animals and microbiomes collaborate and interact in order to promote the health of both of these sides of what we refer to as the holobionts, the animal and the microbiome as a whole. And hopefully we'll be able to use this knowledge and work together with industry to be able to improve production practices, not only for the good of the animals themselves, but also for the growing human population.